गया पूरा। हेलो, गुड मॉर्निंग स्टूडेंट्स। हेलो स्टूडेंट्स, गुड मॉर्निंग। आवाज सुनाई पड़ रहा है? हेलो स्टूडेंट्स, आवाज सुनाई पड़ रहा है? हेलो, एक बार पूछ लीजिए, आवाज सुनाई पड़ रहा है? हेलो स्टूडेंट्स, आवाज सुनाई पड़ रही है? बुक लिख लो। हेलो,
यस सर।
आवाज सुनाई पड़ रही है ना?
यस सर।
ओके, ठीक है, गुड। सो स्टूडेंट्स, today we are going to discuss about the amino acids and a structure of amino acids. In previous classes I have already discussed about that the all proteins are the polymer of alpha amino acids, and you must know that amino acids contain NH2 group as well as carboxyl group COOH group. Alpha amino acids means you can say that depending upon the position of amino group with respect to carboxyl group, amino acids can be classified as alpha, beta, gamma, delta, like that. So NS group attached to next to COOH group, that is called alpha carbon, that is known as alpha amino acids. Now, in previous classes, I have already discussed about the classification of amino acids. That is, first non-essential amino acids, then second essential amino acids. Non-essential amino acids means you can say that those amino acids which can be synthesized in our body is known as non-essential amino acids. For example, you can say that glycine, alanine, symbol of glycine, three, that is gly, for alanine, ala, like that. Similarly, essential amino acids which cannot be synthesized in our body and can be obtained through the diet, known as essential amino acids. For example, valine, leucine, etc., etc. Also, we have all discussed about regarding the acidic, basic, and neutral amino acids. You can say that if the number of carboxylic as well as amino group is equal, that is makes the amino acids neutral. If the amino acids contain more number of carboxylic group as compared to amino acids, that is called acidic. And if number of NH2 group is more than the carboxylic group, that is called basic amino acids. So these are the classification of amino acids. Now we are going to discuss about the properties of amino acids. Dear students, you must know that amino acids are colorless, crystalline solid, having high melting point, and is soluble in water. That amino acids acts as salt due to presence of carboxyl group as well as amino group. That's why they act as salt. Now, you must know that in aqueous solution, carboxyl group present in amino acids can lose a proton, whereas NH2 group can accept a proton to form a dipolar ion known as deuter ion. I again repeat this topic. You must know that in aqueous solution, amino acids, carboxyl group can lose a proton. On the other hand, amino group can accept a proton to form a dipolar ion, which contains both plus and negative charge, plus and minus charge. That's why they form a dipolar ion known as deuter ion, Zwitter, deuter ion. So deuter ion is a dipolar ion which contains both positive and negative charge. For example, listen here you all, dear students. RCHNH2COOH. R आवाज सुनाई पड़ रही है?
Yes, sir.
ओके, RCHNH2COOH है, ध्यान रखना. इसमें से COH का H ए लूज किया और NH2 एक H गेन किया. दैट्स व्हाई यहाँ पर पॉजिटिव चार्ज, यहाँ पर क्या हुआ? नेगेटिव चार्ज. एंड दिस इज कॉल्ड ज्यूटर आयन बिकॉज it is a dipolar ion which contains both here positive and negative charge. That's why this shows amphoteric nature. Amphoteric means they act as both acid and base. That's why their nature is amphoteric in nature. Now, dear students, I am going to discuss about the structure of proteins. You must know that I have already told you that proteins are the polymers of alpha amino acids. Proteins are the polymer of alpha amino acids and are joined to each other with the help of peptide bond, or you can say that peptide linkage. Dear students, keep in mind that very important topic for people. What is peptide linkage or peptide bond? So, peptide linkage can be defined as peptide linkage or peptide bond is an amide formed between COOH group and NH2 group by elimination of water molecule. Thus, the bond so formed is known as peptide bond or peptide linkage. I again repeat it. Peptide bond or peptide linkage is an amide which is formed between COOH group and NH2 group by elimination or by losing of a water molecule called peptide bond or peptide linkage. Here, listen here you all. I have already shown this diagram, NH2CHCOOH and NH2CHCH3, that is called glycine, and here it is alanine. So between glycine and alanine, water molecule that is removed out, minus H2O, and this product so formed must have a CONH group, that is amide linkage, which is called peptide linkage or peptide bond, and is called glyella because glycine, gly, alanine, ala, called glyella. This is the formation of peptide bond or peptide linkage by losing of water molecule between COOH and NH2 group, which is very, very important for people. Now, dear students, I am going to discuss about the classification of proteins. First, on the basis of their molecular shape, you must know that there are two types of proteins. First is called fibrous proteins and second is called glycoprotein. Keep in mind, very important topic. always asked in both question that they differentiate between fibrous protein and glutinous protein. Give at least 2 or 3 difference between these two. You must know that fibrous protein means, you can say that when the polypeptide chains run parallel and held together by hydrogen and disulfide bonds, they form a thread-like structure called fibrous protein, whereas in globular protein, its shape is spherical. So keep in mind, fibrous proteins have linear structure, whereas globular proteins has spherical structure. Secondly, you can say that fibrous proteins are insoluble in water, whereas globular proteins are soluble in water. And third difference, you can say that, for example, keratin, which is found in hair, myosin, that is found in muscle, are the examples of fibrous proteins, whereas insulin, albumin, hemoglobin, that are the example of globular proteins. So this is the basic difference between globular and fibrous proteins. I again repeat it. First point that is of difference, fibrous protein, it is, you can say that, linear structure, whereas globular proteins has a spherical structure. Fibrous protein is on water, where globular protein water. For example, keratin and myosin, that is fibrous protein, and albumin, insulin, hemoglobin, they are globular proteins. Are the basic difference between proteins and globular proteins. Now, I'm going to discuss about the structure and shape of proteins, which can be at 4 different levels. A structure and shape of proteins can be studied in 4 different levels. That is, first primary level, secondary level, tertiary level, and quaternary. Primary, secondary, tertiary, and quaternary. And you must know that each level being more complex than the previous one. हर एक लेवल एक दूसरे से complex होते जाता है. You must know that. So I am going to discuss about the first level, that is primary structure. Dear students, you must know that proteins may have one or more polypeptide chain. Proteins में primary structure में एक या एक से अधिक polypeptide chain हो सकते हैं, and each polypeptide chains have amino acids linked with each other with a specific structure. That polypeptide chains has amino acids which linked with each other with a specific structure. That a specific structure is known as primary structure of proteins. That a specific structure is known as primary structure of proteins, and if any change in the sequence of that group of proteins create a different proteins. अगर उस sequence में कोई हम change कर देते हैं, that generates a different proteins. So that is the concept of primary structure. Now we are going to discuss secondary structure. Secondary structure determines the shape in which a long peptide chain can exist. Secondary structure determines the shape in which a long polypeptide can exist, and they are found to exist in 2 different structures. First is called alpha helix and second is called beta pleated sheet. Again repeat it. Polypeptide chains are found to exist in 2 different structures. First is called alpha helix and second is called beta pleated sheet. First, alpha helix, that is, you can say that these 2 structures arise due to a regular folding of backbone of the polypeptide chain. due to hydrogen bonding between CO and NH group of peptide bond. Now, I'm going to discuss the first alpha helix. In alpha helix, a polypeptide chain found, or it forms, you can say that, all possible hydrogen bonds by twisting into a right-handed screw. Keep in mind always, I again repeat it, in alpha helix, a polypeptide chain forms all possible hydrogen bonds by twisting into a right-handed screw, that is called helix, with NH group of each amino acids residue and hydrogen bonded to CO bond to an adjacent turn of the helix. This formed alpha helix structure. But in beta pleated structure, all the polypeptide bonds or chains are stretched out to nearly maximum. In beta pleated structure, all polypeptide chains are stretched out to nearly maximum extension and then laid side by side, which are held together by intermolecular H bonding. I mean, you can say that intermolecular H bond exists between the beta pleated structure. Third is called tertiary structure. Tertiary structure represents overall folding of polypeptide chains. It represents the folding of polypeptide chains, and it gives rise to two molecular shape, that is called first fibrous and second globular, which I have already discussed here. Tertiary structure gives rise to two different molecular shape. First is called fibrous and second is called globular. And the forces which stabilize the secondary and tertiary structure are hydrogen bonds, disulfide bonds, van der Waals forces, or you can say that intermolecular forces of attraction. These forces stabilize the secondary and tertiary structure. Very, very important. I again repeat it. The forces which stabilize the secondary and tertiary structure are H bonding, disulfide bonds, you can say that, or disulfide linkage, van der Waals forces, as well as electrostatic force of attraction. These forces stabilize the secondary and tertiary structure. Now, last but not least, that is called quaternary structure. Dear students, some proteins are composed of two or more polypeptide chains that are known as subunits. ध्यान रखिएगा. Some proteins are composed of two or more polypeptide chains, that is called subunits, and the spatial arrangements of these subunits with respect to each other is called quaternary structure. I again repeat it, that some proteins are composed of two or more polypeptide chains. That polypeptide chains are called subunits, and the spatial arrangement of these subunits with respect to each other is called quaternary structure. इस सब यूनिट्स का स्पेशल अरेंजमेंट एक दूसरे कंपैरिजन में जो होता है, that is known as quaternary structure. So these are the structure and shape of the, you can say that, proteins. Now, students, we are going to discuss a very, very important topic, that is known as denaturation of proteins. Write the name of the topic, denaturation of proteins. What do you mean by the term denaturation of proteins? Students, you must know that proteins found in a biological system with a unique 3D structure and biological activity, that is called its native protein. Again, repeat it. That protein found in a biological system with a unique 3D structure and biological activity is called a native protein. And when a protein is its native form, is subjected to either physical or chemical change. Physical change means that is change in temperature, or chemical change in the pH. Then what happens if the physical and chemical change is, is observed in proteins? Then you must bonds are disturbed. As a result, globules unfold and helix becomes uncoiled, and protein loses its biological activity. Is called denaturation of proteins. Very, very important topic for people. I again repeat it. Denaturation of proteins. When protein is its native form means you can say that protein is found in biological system with biological activity, that is called its native protein. And when any change is subjected in native protein, that is the change in temperature, then the hydrogen get disturbed, and as a result, globules unfold, helix get uncoiled, and protein loses its biological activity. This phenomenon is known as denaturation of protein. Very, very important for you. For example, you can say that coagulation of egg, curdling of milk, etc. are the example of denaturation of proteins. One important question regarding the denaturation of protein: where does the water present in the egg go after boiling the egg? अगर एग को हम बॉयल करते हैं तो इसके अंदर का पानी कहाँ चल जाता है? Where does water present in chapter called nucleic acid? Nucleic acid, NuCleic, that is called nucleic acid. So, what do you mean by nucleic acid? Dear students, you must know that nucleic acids are the biological polymers which are present in all living cells. Simply, you can say that nucleic acids are biological polymers which are present in all living cells, and these are required for the storage and expression of genetic information. जेनेटिक इंफॉर्मेशन के स्टोर के लिए, एक्सप्रेशन के लिए न्यूक्लिक एसिड्स वेरी वेरी एसेंशियल हम लोग के लिए, एंड दे प्ले ए वाइटल रोल in the transmission of hereditary characters or characteristics, and in, you can say that, biosynthesis of proteins. That acids are, you can say that, very essential and play a role in the transmission of hereditary characteristics and in biosynthesis of proteins, and they control the metabolic activity of all living organism. लिविंग ऑर्गेनिज्म को बायोलॉजिकल एक्टिविटी को भी न्यूक्लिक एसिड कंट्रोल करता है. These are the, you can say that, introductions of nucleic acids. Now I am going to discuss about the chemical composition of nucleic acids. Chemical compositions of nucleic acids. You must know that nucleic acids are the complex and amorphous compounds and are made up of 3 units. Dear students, you all keep in mind that nucleic acids are complex compounds and are made up of 3 units. First, nitrogenous bases. Second, sugar. And third, you can say that phosphoric acid. I again repeat it, that nucleic acids are made up of 3 units. First, nitrogenous bases. Second, sugar. And third, phosphoric acid. First, that is nitrogenous bases. I'm going to discuss very important topic or questions for you people for examination point of view, that you must know that DNA contains 4 bases, namely adenine, guanine, cytosine, and thymine. I again repeat it. DNA contains 4 bases, that is adenine, guanine, cytosine, and thymine, whereas RNA contains adenine, guanine, cytosine, and uracil. This is the basic difference in the DNA and RNA. RNA में थायमीन के बदले यूरासिल हो जाता है. And the 2 types of sugars are obtained by the hydrolysis of nucleic acid. You must know that DNA, RNA, DNA stands for deoxyribonucleic acid, whereas RNA, ribonucleic acid. In DNA molecule, sugar part is beta D 2-deoxyribose. याद रखना, in DNA molecule, sugar part is beta D 2-deoxyribose, whereas in RNA it is beta D ribose. And both of them contains a 5-membered heterocyclic ring and are called pentose sugar. 10 minutes बचा हुआ है, indicate कर रहा है. Dear students, both of them contain a 5-member heterocyclic ring and are called pentose sugar. Now I'm going to discuss here the structure of this ribose and 2-deoxyribose. Listen here, you all. This is the structure of beta D ribose and beta D 2-deoxyribose, in which here the difference of this between 2 is only at this position, which is 5-membered pentose sugar ring, H and 2 position, that is H and OH. In rest all the part it is the same, HOH, HOH, HH, that is different here, HOH, HCH2OH. That is called beta D ribose. Here it is called beta D 2-deoxyribose. These are the structure of this 2, that is ribose and 2-deoxyribose. Now I'm going to discuss here the arrangement of constituent of nucleic acid. I'm going to discuss here the arrangement of constituents in the nucleic acids. इसकी अरेंजमेंट में क्या डिफरेंस है वो थोड़ा जानने की कोशिश किया जाए। डियर स्टूडेंट्स, देयर आर 3 बिल्डिंग ब्लॉक्स इन न्यूक्लिक एसिड्स। आज बता चुके थे पीछे आपको। देयर आर 3 बिल्डिंग ब्लॉक्स इन न्यूक्लिक एसिड, बता चुका आपको, नाइट्रोजनियस बेसेस, सुगर एंड phosphoric acid. A combination of base and sugar is called nucleoside. याद रखिएगा हमेशा। In 3 building blocks, you can say that a combination of base and sugar is called nucleoside. Nucleoside. Similarly, when base, sugar, and phosphates form nucleotides. I again repeat it. Combination of base and sugar, that is called nucleoside, and the combination of base, sugar, and phosphates form nucleotides. While nucleic acids are polynucleotides, which means these are the polymer of nucleotides. In short, you can say that nucleic acids are the polymers of nucleotides, and nucleotides consist of base, sugar, and phosphates. These 3 combine together to form nucleotides. So the nucleic acids are the polymers of nucleotides. Now I'm going to show the structure of nucleic acids that they have already discussed here. The polymer of nucleotides, which can be expressed such that, dear students, base, sugar, phosphate, this do forms nucleotides. When base, sugar, phosphate, base, sugar, like that, it forms a polymer of nucleotides, and this constitutes the structure of nucleic acid, which I already discussed here. Now, second last topic I want to discuss here, the difference between DNA and RNA. Very, very important question for people for examination point of view. Difference between DNA and RNA. First difference, that is, in DNA, pentose sugar present in DNA is 2 beta D 2 deoxyribose, whereas in pentose sugar in RNA it is ribose only. I again repeat it. In DNA, the pentose sugar present is 2 deoxyribose, whereas in RNA it is ribose. Second, I have discussed over here that DNA contains adenine, guanine, cytosine, and thymine, whereas RNA contains 4 bases, that is adenine, guanine, cytosine, and second difference. Third, you can say that DNA is a double chain of polynucleotides. DNA is a double chain of polynucleotides, whereas RNA is a single chain of polynucleotides. DNA डबल चेन होता है पोलिनुक्लियोटाइड्स का, फिर आरएनए सिंगल चेन होता है पोलिनुक्लियोटाइड्स का. Fourth difference you can say that DNA can replicate itself, whereas RNA cannot replicate itself. So these are the difference between DNA and RNA. That is the structural and functional difference between DNA and RNA, which is very, very, very important for you people. Now, last but not least, I am going to discuss about the DNA fingerprints. Dear students, you must know that every individual has unique fingerprints, and that occurs and used for the identification for a long time, but this can be altered by surgery, change hota hai. So a sequence of bases on DNA is also unique for a person, and the information regarding is called DNA fingerprint. इसी का नाम होता है fingerprint. So DNA fingerprinting is now used for many purposes. First, you can say that in forensic laboratories for identification of criminals, nowadays this DNA fingerprint plays a very important role in forensic laboratories for identification of criminals. Second point, you can say that this DNA fingerprint is used to determine the paternity of the individual. Third, you can say that it is also used to identify the dead bodies in an accident for comparing the DNA of parents or children. And last, you can say that DNA fingerprints are used to identify racial groups to rewrite biological evolution. So these are the application of DNA fingerprinting. And last topic, that is biological functions of nucleic acid. Dear students, you must know that DNA is the chemical basis of heredity and regarded as the reserve of genetic information. It is the reserve of the genetic information, and DNA is, simply you can say that, exclusively responsible for maintaining the identity of different species of organisms over millions of years. Here, DNA plays a very important role, and I have already told you a DNA molecule is capable of self-duplication during cell division. And you can say that an important function of nucleic acid is the protein synthesis. I have already discussed here. In the cell, actually proteins are synthesized by various RNA molecules in the cell, but the message for the hello.
Sir, I have a question.
हाँ,
सर कल की टेस्ट का सिलेबस क्या है?
chemical kinetics.